Today I'm going to talk about some of the work I've done over a year, last 1.5 years on blue waters, and also introduce some of the work that we are doing now on blue waters. What I'm basically working on are a lot of signaling proteins, and as you know, that signaling proteins are very important for a variety of diseases. You can name a disease and probably be related to some signaling protein. You can name all forms of cancer would be related to kinases. We're working on and the thing that we really want to understand is that how small molecules can change the behavior of these proteins, and and how do they change the behavior in terms of their conformational preferences. So this is a challenging problem, but it's also a very, very important problem. Again, if you look at all the drugs that are on the market right now, you'll find that roughly 3/4 of them would be targeting some form of a signaling protein, either a kinase or a G-Protein Coupled receptor or other receptors. There's a problem that's ripe for computational investigations, and there are a lot of groups who are working on such problems. And what I'm showing you here is a crystal structures of G-Protein Coupled receptors that were crystallized a few years ago. And the green one is an active form, the orange one is an inactive form. And what we want to do is that we just know these two snapshots really. And what we wanted to do is to connect and find out how these snapshots interconvert into each other, and what are the pathways for their interconversion. Are there intermediate states that we could potentially find which have not been crystallized before? That could give an idea for people to crystallize new forms of these proteins. But again, the problem with these systems is that the size is huge if you're representing it in great atomistic detail. These timelines are a little off, maybe an order of magnitude faster now as compared to what I'm showing you here. But it's still, if you're studying a process that is several milliseconds, you still need, on a standard computer, it will be very, very slow. So what we want to do is we want to develop methods which could accelerate these processes. And this is something that we're trying to run now on blue waters. So I'll give you an example of what we are doing to accelerate these processes. There were a lot of interesting talks yesterday which talk about different methods of accelerating the search of the conformational landscape. What we are using is something called Markov State Models. And if you're not aware of these models, the simple idea is you can think of a very simple reaction, that is A is converting into B. And there are two rates and two states here. And you can go to a much more complex, slightly complex model. Now you have 4 states and you have 16 different rates connecting them. If I know all the rates of interconversion between different species, I can basically predict what will be the dynamics of this protein over time. And a very simple differential equation that you can write. If you just know all the rates and your initial populations, you can propagate this over time. But the problem really is, how do you find all these rates and how do you find all the states that are in between the two snapshots that I've got? To represent this in a very simple way, the question is, how do you get rate? And this is sort of a graph that you would have. All these circles are different states of a protein, and all the lines that are connecting them are the rates in between them. What you can do is that if you're starting from a state that's like this A, you can go to an adjacent state very quickly. So you can run a quick, like a day-long job on a GPU or a CPU node, and you can potentially go from A to B. But if you want to go to rare states, like you're going from A to C, which is a very, very slow process, occurring over hundreds and thousands of microseconds, but you can essentially describe an A to C process simply as jumps which are hundreds of microseconds long. Okay, and this is the central idea in a very simplest form. I'm not going into the mathematical details of this idea, but if you just grasp this thing, that this is a long process that could be described as small jumps, and we can do these jumps over a very, very short periods of time. And I will give you a little demonstration. This is a simple Muller potential. The blue is a minima. The red is high energy states of a protein. For example, you can think of that way. And I want to go from one blue minimum to the other on the, the other corner of this picture. What we are doing is that we are starting from just one structure that we know, and we run a lot of simulations over a 24-hour period, let's say. And then we cluster those conformations. And then we try to find out what are the new states I've discovered today in terms of like from my simulation data. And then I start my new simulations from the newly explored regions of the landscape. These ideas have been implemented in software packages like Copernicus and in MSM Accelerator. What I'm doing on Blue Waters is that we have our own little script that runs through these calculations on Blue Waters, and then we update it. We do a clustering and build a Markov model over a night, and then it just runs over and over again, and then you can fill in the entire landscape. And I'll just show you a very simple picture, a little video of what is this process. So this is what we are doing, and this is what a standard MD trajectory would look like. And you can see that how distributed simulations are running over multiple nodes explore the edges of your conformational landscape much more efficiently than single molecular dynamic simulations that's just sitting in a basin. So you can take if you take any long time scale MD paper that has been published, what you'll probably see is that you have a trajectory that's just sitting at one basin for a very, very long time, and then a sudden thermal fluctuation just takes it over to the other state. But then the problem here is that even if that sudden thermal fluctuation happens once, you need it to happen maybe 10 or 20 times to get some statistics out of that process. Whereas what you're doing here is that because you're starting your simulations in different regions of the landscape that have been explored through unbiased MD, you are crossing over the barrier much, much, much faster than a single MD simulation just sitting in the basin. The total aggregate time in these two figures is same, but you have explored the entire landscape much more efficiently. So this is an order of magnitude faster that way, and you sample the low populated regions well, which is important for understanding the mechanism of conformational change. So we applied this idea to G-Protein Coupled Receptors, and these are, as I said, very critical proteins involved in all forms of sensing. You're drinking a cup of coffee, the caffeine is going to bind a G-Protein Coupled Receptor. You probably take 1/3 of all medicines are going to hit this protein. So it's a very, very important target, and that's why Brian Kobylka and Bob Lefkowitz got the Nobel Prize 2012 for finding out this crystal structure. Now this crystal structure is very interesting. It's a big discovery, but again, as I said, it's a single snapshot of a protein. It's not a protein in motion. You don't understand what are the functions of this protein, how it is interconverting, how it is doing its function. What we can do with the large scale computation, especially with Blue Waters and all the GPUs that are available to us, is that we can sample the conformation landscape, get all the snapshots of this protein. And I'll just show you a very quick picture. This is an inactive state, and these are the 3 regions that are important. You see how inactive to active transition happens. Little flip of the ring, twist of this little helix, and then this purple helix moving out, right? So inactive to active, as you're seeing, there's a big change here. And these are the key parameters that are, these are the key residues that have been identified, again, over like 40 to 50 years of experimentation. So people have done a lot of mutagenesis to identify each of these little changes in the protein are important for its function. But what you're doing in silico, starting from a crystal structure, is you can learn these things within a few months' time, which have taken decades to do. And this is just a long MD trajectory that is generated from stitching together all those short MD simulations that we have done. Again, you can see in the presence of an activating ligand, the protein activates. Without a ligand, it just stays in basal state. And so you can get all these correct biological phenomena from this. One other important thing is that you can also take a very long simulation on the exact same system. And we were very lucky that D.E. Shaw had done all those simulations of beta 2AR, which is the same protein as we are simulating. And we can compare those long trajectories with our very, very short trajectories. And you can look at the autocorrelation functions of different degrees of freedom, and you find the values are the same. So these short trajectories are not giving you anything less than what a long trajectory would give. So that's exciting. This is something, and then we can apply it to other systems and problems. One other interesting thing about this is that you can do these simulations now in the presence of different drugs, and you see this conformational landscape where there's one drug which prefers a particular pathway, there's another drug which prefers another pathway, which was not visible when the other drug was present. So you're seeing this structural or conformational plasticity where the drugs can distort what type of conformational pathways the protein goes through when it activates or deactivates. But we can do something more than that. What we did is that we took these intermediate states, and what I'm showing you in this figure is an activation progress variable going from 0 to 1. And intermediate structures that we found from our simulations, we are doing docking of a large portion of the zinc library of molecules, and we are finding out new molecules that have never been that you cannot find if you just had the active and the inactive crystal structure. So these new structures are giving you new classes of compounds that we have not been able to find just by using the crystal structures, although experimentally you can show that these compounds have an effect. Okay, so that's interesting. This was again on Nature Chemistry. This is more recent work done on blue waters in the last 3 months. So this is a protein called Calmodulin. It's a calcium modulating protein, very important. And as you all know that our bodies maintain big gradients of ions across cell membrane, and this is a protein that sort of that binds calcium and then initiates signaling to do a variety of processes in our body. The yellow one is a protein without calcium, and then the orange one or brown one is with calcium. And you see this huge conformational change in this protein happens as calcium binds it. And what we are able to do is that, again, I'll show you a little more close-up. What happens is that as calcium binds, all these charge residues that are holding calcium ions, they just move away when the calcium is not there. Similarly, what it does, calcium binding does, is it exposes hydrophobic surfaces on this protein, which allow it to bind different proteins. So what we were able to find is that how we can look at the two crystal structures that are available, apo and a holo-like, but now we are finding intermediate conformations of these hydrophobic surfaces that have never been crystallized. Okay, and that's very interesting because these could be a target for new types of drugs where you're binding drugs on the hydrophobic interface and thereby stopping its interaction with a particular binding partner. And we were able to find these interfaces. And this one particular interface, the intermediate one that I'm showing, is also been crystallized in the presence of a drug molecule. So you're finding these new structures that have not been crystallized, and this has some potential for designing new drugs. Similarly, we find, as I said, a lot of different types of topology of the hydrophobic interface, which are chemically, aesthetically distinct. And again, this shows you a potential where there's been decades of studies on this protein. Again, you have crystallized a lot of different structures, but you have not found all of them. And whereas what you're doing with computations is that you're finding all these different conformations. The good thing about these predictions is that this is a conformational landscape. You don't have to worry about the axes here. But the point is that these white dots that I'm showing you are different crystal structures that have been crystallized. So we are starting from just 1 or 2 structures that are available, and then finding out all the crystal structures that have been reported till now, and something extra. So that sort of validates that the crystal structures, the structures that we're getting from MD are actually real structures. So they are not not just random predictions. So that gives some level of confidence in our work. We have also validated this work using NMR experiments. So this again matches very well. The kinetics is also right. Okay, and then this is final thing that I'm doing. This is a new project that we're working on. Is slightly cut. Is like we're working on molecular design of drought-resistant plants now. This is a figure of past droughts that happen. Every few hundred years, you have these serious droughts in the United States. If you look at the current drought conditions in the country, it's gone only worse. This was August, so last year. But now it's even worse in California. The entire California is under this exceptional drought. So entire Southwest is sort of suffering. and the future does not look that bright. The probability of having a serious drought in the southwestern states is very high. So there's a lot of interest now from agricultural scientists, from plant biologists, to work on plants and design plants that are drought resistant. And what we are doing is we're trying to study molecular processes and cellular signaling to study these things in plant proteins. And there are many gaps in this area because this is new. There's not much of computational plant biology that exists. So what we are trying to do is to fill in all the gaps that are there and find a lot of challenging problems here. What I'm particularly working on right now is plant development through steroid signaling. We are trying to understand this particular receptor, which is called the BRE1 receptor. It's important for plant growth. So a steroid hormone is going to bind this receptor, and then it recruits other receptors and then signals the plants to grow. Can we have interventions in this process of signaling such that it promotes plant growth? What I have done so far is on blue waters is that we are simulating the binding of the steroid to the receptors, and we are trying to see if we can have mutations in the plant that could inhibit the binding or make it more attractive to the plant or bind it much, much strongly. And obviously, there is very little structural information in this field. So anything that we can do from computations and provide structural information is also a great help to this field. This is my initial work. What we found were, I'm not showing you a lot of results, so I'll wrap up quickly, is that this is the receptor. We did a lot of simulations on this on blue waters. And what we found is that for binding of this particular steroid hormone, these few residues are really, really important. And if we can do mutations of these residues to charge residues like glutamate, we can potentially inhibit the binding. And so these results are there, but I don't want to go into them because they're still under preparation, sort of. The paper is under preparation. But we have experiments now, and I'm really excited about this. That's what I'm showing you this figure, is that we have a control that is that corner, and this is the plant that we have from the mutation. And this grows much, much bigger. So there's a lot of potential to do these things at Illinois. And so this is with Steve Huber at UIUC and USDA. And this is my last slide. What we are trying to do on blue waters is to do some type of computational plant engineering, not just at the molecular level, but take it to a cellular level or a system level or an ecosystem level. And we have different professors at the University of Illinois, and we're coming together to have this computational plant platform that we can use HPC for. So I'm at the foundational level at the molecular level, but now we are also trying to work to connect these different levels together. And with that, I would like to thank Blue Waters, Alexander Moffat and Zahra Shamshi, who is shown on the right-hand side of this figure. They did all the work. And some of the initial work I showed on GPCR was done with Professor Vijay Pandey. So I would also like to acknowledge him. Thank you, and I'm happy to take any questions.
Right. Thank you, speaker.
Yeah.
Time for questions.
Especially, so how did you obtain the hydrophobicity from the calcium? You said you're using calcium to determine the hydrophobic surfaces?
No, so the calcium is actually binding to the protein, and when it binds, it changes the conformation of the protein. So the hydrophobic surfaces that were collapsed now are open.
But the method you're using to determine the hydrophobicity is?
The method that you can use for, you can use any standard hydropathy scale, and that would be one standard answer. But you can also do some other things like, water fluctuation around the surface and see, what is the net hydrophobic.